Okay, um, we want to pick up where we left off talking about immune defense. Our adaptive or specific immunity is the last line of defense between your body and the invasion of pathogens. It protects against infectious agents and abnormal body cells, and it amplifies the inflammatory response. It activates complement, and it has to be primed by initial exposure to a specific foreign substance in order to be effective, and this priming takes a little bit of time. It's specific because it recognizes and targets one particular collection of molecules. These are what are called antigens. They're basically ID tags that indicate that the invading organism is not a member of your body's tissues, and as a result will be recognized as not self. Remember that specific immunity does not differentiate based on value judgments. It simply discriminates on whether or not a particular cell is identified as belonging to the body or not belonging to the body. And the attack is based on that criteria. It's systemic, so it's not restricted to the initial site of the infection, and it has memory. It basically can attack known antigens the next time they show up as a result of the production of special cells called memory cells during the course of the attack. It has two separate arms: humoral or antibody-mediated immunity and cell-mediated immunity. Humoral immunity is mediated by B cells, and they create antibodies which are basically magic bullets that circulate in the body fluid and will bind only to a cell or a tissue that displays that particular antigen. So it can act at a great distance from its initial site of production. They bind temporarily to the target cell and inactivate it and mark it for destruction either by phagocytes or by complement or by a phenomenon known as immunoprecipitation, in which cells of a particular pathogen are linked to each other, and at a certain point the molecular weight gets so large that the entire complex falls out of solution and becomes easy pickings for a wandering macrophage. Cellular immunity is mediated by lymphocytes that act against the target cell. They do this directly by killing the infected tissue and indirectly by releasing chemicals that enhance the inflammatory response or activating other lymphocytes or macrophages. An antigen is simply a substance that identifies a particular cell as being a member of your body's tissues or not. They're sort of like ID tags. Substances that can mobilize adaptive defenses and provoke an immune response are classified as antigens. Targets of all adaptive immune responses are centered around this antigen-antibody recognition. Most of these are large, complex molecules that are not normally found in the body. For instance, in an invading virus or a bacteria, or even something as innocuous as a pollen grain, we can display a collection of molecules that's not found on any of your own tissues, and the result then is to mount an attack against an organism so tagged. Complete antigens have important functional properties. They're immunogenetic, meaning that they can stimulate and particular lymphocytes to proliferate. Not every foreign collection of molecules has antigenicity. It takes this particular collection of atoms bound to each other in a particular way in order to alert the immune system to the presence of this foreign invader. It also has reactivity, which is the ability to react with activated lymphocytes and antibodies released by immunogenic reactions. So just binding is not enough. It also has to spur now a response after the binding. Examples include foreign proteins, polysaccharides, lipids, and nucleic acids, or collections of one or more of each of these. Haptens are incomplete antigens. They're not immunogenic by themselves, but they become immunogenic when they're bound to larger molecules. Examples include peptides, nucleotides, and hormones, just to name a few. Again, they may be immunogenic if they're attached to body proteins, and this combination is marked as foreign and targeted now by a particular antibody. It can cause the immune system to mount an attack that is directed at the antigen, but can also harm your own tissue in collateral damage. And this is the hallmark of what we call hypersensitivity. We'll talk a little bit about that. Examples include poison ivy, animal dander, detergents and cosmetics, but again, it's not limited to these classes. There are a wide variety of haptens that you can get from a wide variety of sources. Only certain parts of an antigen are immunogenic, and these are called determinants. Antibodies and lymphocytes have receptors that bind to them as enzymes bind to substrate. So this is sort of a lock and key interaction. Again, specificity is very important here. Most naturally occurring antigens have numerous determinants that mobilize several different types of lymphocyte populations and form different kinds of antibodies against it. So when a foreign invader comes in, he may have one or more collection of molecules that could activate a particular B or T cell clone and have them generate an attack. There are large chemically simple molecules that foreign bodies often bring in that have little or no immunogenicity, and as a result they'll slip by the immune system. And this is actually a strategy that's employed by certain pathogens to avoid immune detection, which is to have an antigen display that is not immunogenic. And so here you can see an example of a particular antigen and the determinants on that antigen, and how different antibodies, different magic bullets, can each recognize this particular epitope. Now, one of the things I want you to notice in this drawing is that each antibody has at least 2 antigen binding sites. And the result of this is that one of the methods by which an antibody can render a pathogen harmless is by binding individual pathogen cells or virus particles or what have you to each other and form a complex that's so large that the pathogen is effectively immobilized and is easily destroyed by wandering macrophage. So here you can see how that might work. So if you had another antigen nearby, it could be bound to this complex, and in turn another antibody could bind to that and bind to another antigen, and so forth. And eventually the complex gets so large that it's no longer soluble. Self-antigens are found on the surfaces of all the cells in our bodies, and they're not antigenic to our own immune system, but they can be antigenic in transfusion or in grafts. Basically, when the immune system is initially formed, there are a couple of criteria that have to be met. One criteria is that it has to be tolerant of our body's own antigens, meaning it will pass them by and not attack them. If not, those potential lymphocyte clones are destroyed or rendered basically inoperable. They exist, but they are not capable of mounting an immune response. This is something called anergy. They also have to be capable of recognizing our MHC proteins and recognizing foreign antigens. And so they have to be what's called immunocompetent. So tolerance and the ability to mount an immune response when exposed to a foreign antigen, 2 very important criteria that eventually form the large population of cells that are genetically distinct that are going to make up your lymphocyte population. Examples of these include major histocompatibility glycoproteins, which are coded by genes of the MHC complex and are unique from individual to individual with the exception of identical twins. And they have a groove that holds the self or the foreign antigen kind of like an hors d'oeuvre on an hors d'oeuvre plate. Okay? So the MHC is going to have something in its groove, something on its plate. And if that something is one of our own antigens, a lymphocyte that is both tolerant and immunogenic will be able to recognize that and not destroy that particular antigen display or the cell that generates it. At the same time, it has to be capable of recognizing your MHC proteins in combination with an unusual antigen, something that's brought in from the outside. And the way this happens is through what are called antigen-presenting cells, which engulf foreign particles, break them up using lysosomes as a method of degrading the invading organism, and then presenting those antigens on the surface in combination with the MHC protein for potential recognition by a wandering B or T cell. And that's what spurs the immune system to generate its specific attack. Lymphocytes only bind antigens on MHC proteins, and as a result, they then mount their attack. Okay? So the adaptive immune system cells come in three types. There's two types of lymphocytes, B and T lymphocytes. The B lymphocytes, remember, are responsible for humoral immunity, while the T lymphocytes are responsible for cell-mediated immunity. And antigen-presenting cells that don't respond to antigens, but engulf foreign invaders, chop them up, and display those antigens on their surface in order to alert the immune system to their presence. So there's 5 general steps in lymphocyte development, maturation, and activation. First, we have to form all the cells that are going to make up the immune system, and this happens in the red bone marrow. Remember, in the adult, that the red bone marrow is found in the bony thorax and the heads of the femur. Then they have to mature. Then we have to seed secondary lymphoid organs and circulation once we've been properly educated in either the bone marrow or the thymus. And then we have to encounter antigens and activate ourselves, at which point we proliferate and differentiate and mount the attack against the foreign invader. Maturation is basically the education of these lymphocytes. They go from naive to mature. B cells do this in the bone marrow, T cells do this in the thymus. Immunocompetence is essentially the property whereby a lymphocyte can recognize one specific antigen by binding to it. B or T cells display unique receptors on their surface when they get immunity, and these receptors are only capable of binding a single antigen. And they're also self-tolerant, meaning that the lymphocytes are unresponsive to our own antigen. So they essentially have to have the capability of making a receptor and or an antibody that is going to recognize a particular foreign antigen, but they also have to be tolerant of our own antigen display. Now, one of the riddles in biology for the longest time was how in the world it was possible to make billions of different lymphocytes that have all these particular specificities with the amount of genetic material that's found in the nucleus of a typical human cell. And it turns out that the lymphocytes are the exception to the rule that states that if we were to look at the DNA in every cell in the body, for its total sequence from front to back, that it would be the same from cell type to cell type. For instance, if we take the DNA from a nerve cell and the DNA from a muscle cell and a bone cell and a connective tissue cell and epithelial cell, and we sequence it front to back, what we would find is that the sequence is basically the same, okay, pretty much identical. The difference in cell morphology and cell behavior comes from the fact that these cells read their genetic material differently as a result of chemical modifications that take place on particular genes that render them inactive for particular cell types, so that essentially what causes a muscle cell to look and act like a muscle cell is that it's reading from that segment of the DNA which you could think of as the muscle handbook. It's able to make actin and myosin and all the other proteins that are associated with the sarcomere, for instance. and it doesn't really make very many of the proteins that are involved in other processes, such as um, the synthesis of enzymes that control things like the urea cycle. Okay, that's not a big deal in a muscle cell. Why that difference? Well, because we've shut off those regions of the DNA, even though the material, the genetic material is still there, the cell isn't capable of utilizing it. Okay. Now, the exception to that is the lymphocyte. If we were to take the genetic material from a lymphocyte and compare it to the genetic material of other cell types, like a bone cell or a muscle cell or a nerve cell, we would find that there are profound genetic differences within particular locations of the DNA. In T cells, we would find massive differences in the genes that code for the T cell receptors, and massive differences in the B cells in the regions that code for their antibodies. And the reason that this happens is because during the formation of B and T cells, while you're still in your mother's uterus, what happens is that segments of DNA that will eventually contribute to a functioning T cell receptor or antibody gene are picked kind of at random and combined, just like pieces of a Lego set, in the hopes of making a functional antibody or T cell receptor. The selection of these segments of DNA is really kind of up to chance. And furthermore, when these segments are assembled, what happens is that little nucleotide sequences at the ends or the beginnings of these segments can be either added or subtracted in the process of joining these segments together. And so what you end up with as a result of this sort of deck of cards of DNA that can make a T cell receptor or an antibody is the potential to make billions of different unique protein coding genes from a subset of genetic chunks that you recombined during the formation of the mature lymphocyte population. Okay, so that's the answer to the riddle of how this happens. And I'll show a little video that kind of talks a little bit about how this works.
Human cells do not have enough DNA to have separate genes for each antibody molecule. Instead, different segments of DNA can be mixed and matched to form different antibodies. Light chains are made from V and J segments in the variable region and a constant region segment, whereas heavy chains are made from V, D, and J segments in the variable region and one of 5 different constant region segments. As the lymphocyte divides during maturation, its genes are rearranged. DNA sequences are assembled by the random selection of V and J gene segments for the light chains and V, D, and J segments for the heavy chains. The constant region of the molecule is encoded by a single C or constant gene segment for light chains and 5 different constant regions for heavy chains. The rearranged DNA in the mature B cell is transcribed into messenger RNA and translated into the light chains of the antibody. DNA sequences for the variable regions of each heavy chain are assembled by the random selection of a V gene segment, a D gene segment, and a J gene segment. The genes for the constant regions of heavy H chains have Greek letters, designating IgG, gamma, IgM, mu, IgA, alpha, IgE, epsilon, and IgD, delta. The DNA in the mature B cell is transcribed and translated into the heavy chain protein. The light chains are then attached to the heavy chains to form the functional antibody. Because of the large number of ways in which the genes can be rearranged, many different antibodies can be produced.
Okay, T cells mature in the thymus under negative and positive selection pressure. They basically have to pass a couple of tests in order to become members of the fully functioning immune system. In positive selection, we pick out T cells capable of recognizing self-MHC proteins. If they can't recognize these proteins, they're destroyed by programmed cell death, which is a process known as apoptosis. This is essentially the flipping of an internal biochemical switch that causes the cell to commit suicide. This is what we call programmed cell death. In negative selection, we prompt apoptosis of T cells that bind to self-antigens that are displayed by the self-MHC. So we don't want these T cells to bind to and recognize our own antigens. We want them to tolerate our own antigens. So if they bind and recognize these particular self-antigens, they're going to be rendered either inactive or they're going to be destroyed. And this ensures self-tolerance. So this is just a diagram showing you how this all works. This is the sequence of events front to back. So we start out with B and T lymphocyte precursors in the red marrow, and the B cell precursors will migrate to the red marrow and begin the process of maturing. Okay, these are the cell populations they're eventually going to make are circulating antibodies. Other precursors, the T cell precursors, migrate to the thymus, where they become immunocompetent and self-tolerant. The next thing that happens is that once these cells have matured in either the bone marrow or the thymus, they migrate out and they populate lymphoid organs around the bodies. These are things like lymph nodes, the spleen, the liver, the Peyer's patches. There's lymphoid organs all over the body, and they tend to be stationed in parts of the body that are likely to encounter a pathogen. Okay, so they tend to be near or around areas of the body that are open to the environment. The next thing that happens is that if they encounter a particular antigen to which they can bind, those particular lymphocyte clones that recognize that antigen as a result of interacting with them either directly or on the surface of an antigen-presenting cell will begin to proliferate. Okay, this is the response to the infection. This proliferation will eventually lead to the production of, in the case of B cells, circulating antibody that can bind antigen and eventually find its target, just like a magic bullet would. Or in the case of T cells, lymphocytes that will actually go out, enter the circulation, and be transported to the area of the body where the infected or diseased tissue is located and directly destroy it. Okay, so that's the difference between humoral immunity and cell-mediated immunity. Humoral immunity, again, is brought about by the production of antibodies that can act at a distance, while cell-mediated immunity, the lymphocyte actually has to show up where the diseased tissue is in order to destroy it, to render it inactive and inert. And so here you can see how this positive and negative selection works when we're generating our T cell population. This is what's occurring here in the thymus. Okay, the thymus, remember, is a lymphoid organ that sits in front of the heart and behind the sternum. So the T cells have to recognize self-major histocompatibility proteins. This is the hors d'oeuvre tray upon which the antigens will sit. Okay, so here you can see an antigen-presenting cell and one of our own antigens on the hors d'oeuvre tray, and here you can see a developing T cell. If we do not bind this collection of molecules, we're going to be rendered inactive or we're going to be destroyed. Why would this be? Well, if we can't recognize an MHC protein, there's no mechanism whereby that particular lymphocyte could defend us against disease, right? Because that recognition involves binding both the hors d'oeuvre tray and the hors d'oeuvre in this case. So if you can't recognize the hors d'oeuvre tray, you're out. However, if you recognize the MHC protein, you're going to be promoted on to the next round of selection. Okay, so recognizing a self-MHC allows you to survive, and these survivors then proceed to the negative selection. If this population of cells recognizes a self-antigen, they're going to be rendered either inactive, as that's a process called anergy, or they're going to be destroyed in the process of apoptosis. And so what you're left with is a population of cells that's tolerant of our own antigens, but capable of recognizing and destroying foreign antigens as long as they're presented in combination with the MHC proteins. So this process eliminates self-reactive T cells that could cause autoimmune disorders. Failure to recognize self-antigens results in survival and continued maturation. Now, very important point here, which is that some of these cells aren't destroyed, they're simply rendered dormant or inactive, okay, in a process called anergy. And defects in this anergy may be responsible in some cases for a cluster of diseases that we call autoimmune disorders, in which your immune system suddenly turns and attacks your body's own tissues. In the event that these cells that are rendered incapable of responding to an antigen display, if they were to activate somehow by some other mechanism, then they could turn around and attack your body's own tissues. The other method by which these autoimmune disorders can manifest themselves is through mutation in the lymphocyte population over the course of your lifespan, in which you may produce an abnormal T cell receptor or antibody that could in turn recognize one of your body's own antigens and destroy it, or mutation in one of your body's own tissues that could result in the display of an antigen that wasn't there during this selection process, and as a result, your immune system would then go out and destroy those particular cells. And then there's probably the most frequent mechanism whereby this occurs, which is exposure to a cross-reacting antigen. So let's take an example. Let's say that this represents a pathogen, and on this pathogen is a particular antigen. I'll just indicate it by these little peaks here. And let's say you get exposed to this antigen during the course of your activities of daily living, and you now generate an antibody against that particular antigen. Okay, so here comes your antibody. Here's your magic bullet. Okay, and you bind and destroy everything with this antigen display very effectively. Okay, well that's great. Okay, so this guy gets aced out. Okay, he is no longer going to be a threat to the body. He's gone. But the problem now is that we have a whole bunch of this antibody kind of hanging around with nothing to do. Okay, well, if there happens to be on one of your body's own cells, let's draw one of your body's own cells here, an antigen. So let's say this is the nucleus. Okay, there's the DNA. And then here's one of your body's own antigens. Let's say that the antigen on your own body is similar but not identical to the antigen that was present on the pathogen. What can happen is that this antibody could somewhat recognize and bind to this self-antigen just a little bit, okay? Not enough maybe to stay bound for a long period of time, but maybe for just a little while, okay? Maybe this binds at about 10% of the strength that it binds its original antigen. But what you have to remember is that now there's a lot of this antibody around, right? We've made tons and tons of it as a result of exposure to our initial cross-reacting antigen. So you could imagine that as soon as, say, this particular antibody lets go, there's another one coming right in behind it that can recognize this antigen. And what can this result in? This can result in an attack on your body's own tissues. Okay? And that seems to be one of the most prevalent mechanisms whereby autoimmunity occurs. But one last mechanism that can produce autoimmunity is to breach something called the blood tissue barrier. There are certain organs in the body that have a property known as immune privilege. Basically, what that means is that They're isolated from the formed elements of blood, and as a result, lymphocytes, the B cells, the T cells, never see those tissues. Okay, examples of tissues in the body with immune privilege include the corneas of your eyes, your liver cells, your tissue of the central nervous system, and your urogenital system. Particularly here, we're talking about in men, the gametes. They're isolated from the blood in the blood-testis barrier, and in the female reproductive system, the corona radiata and zona pellucida that surrounds the oocyte as it's released from the follicle, the ovary, serves as a barrier between the woman's immune system and the gamete that she's produced. In the event that that is breached, what will happen is that you will make antibodies against those tissues. And as an example, in men that have vasectomies, one of the things that they find after the procedure is because we've broken now the blood-testis barrier as a result of the surgery, we now generate antibodies against our own sperm. And this can confound attempts to try and reverse the procedure. Sometimes people have vasectomies and they decide they want to have kids later on, and they try to reverse the procedure, and sometimes it's not successful, and that in part is due to this phenomenon. Okay, so autoimmunity. Okay, B cells mature in the bone marrow. That's where they get their name from. They're positively selected if they successfully make antigen receptors. Those that are self-reactive are eliminated by apoptosis in a process called clonal deletion. And that's similar to the way the T cells are eliminated. Immunocompetent B and T cells not yet exposed to antigens are called naive. These are exported from the lymphoid organs, the bone marrow and the thymus namely, to seed secondary lymphoid organs such as the lymph nodes and the spleen. This increases the encounter with an antigen, increases the ability of these cells to see foreign particles if they show up. Lymph nodes are basically little pea-shaped organs that are stationed all around the body. You find tons of them in the cervical, the axillary, the inguinal, the popliteal, and the mediastinal region. And they're there because these serve sort of as gateways from organs that are likely to encounter pathogens to the rest of the body. Essentially what they do is they filter the interstitial fluid and screen it for the presence of any infective organism. And if they find it, the cells that are in the lymphoid organ will engulf that pathogen, chop it up into little bits, and display those little bits on the cell surface in antigen presentation, and that will serve to alert the immune system to the presence of the pathogen. So, what happens when we have an antigen that comes in from the outside, let's say on a disease-causing organism? A process called clonal selection ensues. The naive lymphocyte's first encounter with an antigen causes it to be selected for further development. Basically, if a stranger shows up, a disease-causing entity, a virus or a bacteria, with a foreign antigen on its surface, what will happen is that the naive lymphocyte will interact now for the first time with this particular antigen. If there is a recognition between the lymphocyte and the antigen display, the lymphocyte will complete its differentiation. The activated lymphocyte will proliferate and make identical clones of this particular lymphocyte, this particular cell. So every cell that results as a process of this proliferation will have the exact same genetic sequence as the original, and that means that they'll produce the exact same antibodies and the exact same T cell receptors, if we're talking about a T cell, as the original. So what happens now is you get large numbers of these particular antibody producing or T cell lymphocytes. Most clones end up as effector cells to battle the infection, but in the course of battling the infection, some of them are chosen to become memory cells. These are able to respond to the same antigen more quickly the next time it shows up. The B and T memory cells and the effector T cells will circulate throughout the body continuously. And what will happen as a result of successfully fighting this infection is that now, as a result of the production of these memory cells, you'll have the ability the next time that same pathogen shows up with that exact same antigen display to effectively knock it out before it gets a chance to make you sick. And this is why very often individuals who were sickly as children as a result of infectious disease, if they've lived through childhood, they tend to be relatively healthy as adults because they have very seasoned immune systems that have seen a lot of antigen displays before.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So where does this diversity come from? And we've actually already talked about this a little bit. Genes determine which foreign substances the immune system recognizes. Immune cell receptors are the result of acquired knowledge of microbes that they encounter in the environment. Lymphocytes make up to 1 billion different types of antigen receptors, coded for by about 25,000 genes. The reason that we can make 1 billion different specificities is because these gene segments are shuffled by somatic recombination, as we showed in the earlier video. And so this gives us the capability now of recognizing virtually any antigen that shows up that is not similar to our own. Okay? Antigen-presenting cells help facilitate this process. They engulf antigens and present the fragments to T cells for recognition. There are several different types of APCs. Dendritic cells are found in connective tissue and in epidermis. Macrophages are found in connective tissue and in lymphoid organs. And of course, B cells are found throughout the body. They're found in lymphoid organs. They're found in the circulation. They're found in interstitial fluid. They're found in a lot of different places. Dendritic cells phagocytize pathogens and enter lymphatics to present the antigens to T cells contained in the lymph node. They're the most effective antigen presenter in the body, and they're a key link between innate and adaptive immunity. Remember that in innate immunity, we use the same mode of attack regardless of the identity of the pathogen. And in adaptive immunity, we sculpt the attack particularly for that individual strain of virus or bacteria or what have you. Macrophages are widespread in lymphoid organs and connective tissue, and they can activate naive T cells as well. They present antigens to T cells to activate themselves into phagocytes that can secrete bactericidal chemicals. And here you can see an example of a dendritic cell. This is taken with an SEM. This is about 1,000 times larger than it normally would appear. But again, what you notice here are all these pseudopods. Okay, and what is this cell's job? To engulf things and to present them on their surface so that they can activate the T cell population. B cells do not activate naive T cells, but they present antigens to helper Ts to help their own activation. So in summary, lymphocytes, antigen-presenting cells, and antigens identify and destroy non-self substances. This depends upon the ability of the cells to recognize the antigens by binding to them and to communicate with one another so that the whole system mounts a specific response. B cells are activated when antigens bind to their surface receptors and cross-link them. The receptor-mediated endocytosis of the cross-linked antigen receptor complexes results in clonal selection that leads to proliferation and differentiation into effector cells. Most clone cells become plasma cells that are involved in making tons of antibody. They secrete antibodies at a rate of 2,000 molecules a second for 4 to 5 days, and then they die off. The antibodies can circulate in the blood or the lymph and bind to free antigens, marking them for destruction by innate or adaptive mechanisms. Clone cells that don't become plasma cells become memory cells and provide immunological memory for your entire lifetime. They mount an immediate response to future exposure to the same antigen. The primary immune response is the cell proliferation and differentiation that happens upon first exposure. There's a lag period of a little under a week, and peak levels of plasma antibodies are reached in about 10 days. The antibody levels then decline. However, the second time that you're exposed to that same antigen, the secondary immune response is much more robust. Re-exposure to the same antigen gives a faster, prolonged, more effective response. The sensitized memory cells respond within hours, and the antibody levels peak in 2 or 3 days at much higher levels, and the antibodies also bind with greater affinity. The antibody level can remain high for weeks to months, and as a result, you really don't get sick the second time around. And so here we see humoral immunity at its outset. Okay, so we get our primary response. We have our B cell encounter with the first antigen. These B cell receptors cluster at endocytose. Okay, and now the antigen is going to activate this particular B cell. Okay, what will happen as a result of this is that the B cell will display some of this antigen on its surface and interact with something called a helper T cell that has the same specificity. And that helper T cell will produce interleukins that will cause even greater proliferation of this B cell population so that it can proliferate and go out and form a clone. Most of these activated B cells become plasma cells. Notice that they have an extensive endoplasmic reticulum because they're in the business of producing large amounts of protein in order to battle the infection. While a small percentage of them will go on to become memory B cells that will remain in the body throughout that individual's lifetime and respond to a subsequent secondary exposure with a massive production of clones, which will in turn produce tons of antibody and render that pathogen incapable of making you sick. In the primary response, we still make these plasma cells, but we don't make nearly as many of them, and they don't produce nearly as much antibody. So the first time you're exposed to a pathogen, it takes a while to knock the disease back naturally. But in the course of doing this, these memory cells get selected and they get stored in the body, and then the next time the pathogen shows up, you're able to wipe it out tout de suite. And so here you can see a look at the primary and secondary response. Notice that in the primary response we have an initial exposure to an antigen, and antibodies begin to peak between 7 and 10 days after exposure. Okay, and then we wipe out that particular pathogen. The second time we're exposed to that same antigen, look at the difference in the antibody production. Okay, it peaks way up, and it happens way sooner. Okay, within 2 or 3 days, and we knock out the pathogen before it ever gets a chance to make us sick. If we're exposed to a different antigen, look what will happen. We'll get now a primary immune response to the second antigen, and then if that second antigen were to show up again, we would have a very robust response to antigen B. Okay, so this is one of the hallmarks of specific immunity is memory. Okay, the ability to the second time that antigen appears, knock it out. When B cells encounter antigens and produce specific antibodies against them, there's two types of active humoral immunity. What we're looking at here is how it is that we generate the defense. Okay, what are some different modes whereby the defense is generated? So there's two types of active humoral immunity. In naturally acquired immunity, we have a response to an exposure as a result of our activities of daily living. In artificially acquired immunity, we are exposed as a result of an injection. Okay, so that might be, for instance, a vaccine. Vaccines are composed either of dead or attenuated pathogens, or these days, really, they're composed of protein components of the pathogen that are produced in pure form via genetic engineering. We really don't use dead or attenuated vaccines that much anymore. We generally try to find genes that code for coat proteins and clone them into harmless bacteria, produce massive amounts of that particular antigen, and use that as a component of the vaccine. And this spares us the symptoms of the primary response, and it also provides antigenic determinants that are immunogenic and reactive. And so what happens is you have this primary response um, against this antigen, which isn't in this case on a pathogen, and as a result, the next time that the pathogen shows up with the same antigen, you're able to knock it out. This can cause illness trying to vaccinate. This can cause the illness that we're trying to vaccinate against, and it can also cause allergic responses. Essentially, what they're talking about here is in the use of the dead or the attenuated pathogen. Okay, again, we don't use that much anymore. Kind of blot this out of your mind because this isn't the composition of most vaccines these days. Essentially, adverse events to immunization come primarily from hypersensitivity to what could be any antigen. And that's going to vary from individual to individual depending on the composition of their immune system. And so what you're having there is not the disease per se, you're having an allergic reaction to the vaccine. And so when you hear about people getting sick as a result of getting the vaccine, they're not generally getting sick with the disease that the vaccine is aimed against. What they're having is a hypersensitivity reaction. Naked DNA and oral vaccines are another approach that allow us to prepare the immune system for the presentation of a particular antigen. Now, how do these work? Well, essentially, what you do with these naked DNA vaccines is that the DNA gets taken up by lymphocytes and it gets expressed and it codes for an antigen that ends up on the surface of that particular lymphocyte. And now, what will happen, lymphocyte or macrophage or antigen presenting cell, you take your pick. What will happen now is that antigen is now in the mix. That antigen's in your body, it's being exposed to the immune system. And what will happen is that you'll mount an immune response against that antigen, which was produced as a result of expression of that chunk of DNA in that antigen presenting cell. And the result then will be an immune system that's now prepared for the next time that particular antigen shows up on the surface of the pathogen. Passive humoral immunity is where we get an antibody injection in the body. B cells are not challenged by antigens in this case, and there's no immunological memory. And the protection ends when the antibodies break down. And there's two types of passive immunity. There's naturally acquired immunity, such as the antibodies that are delivered to the fetus through the placenta or to the infant through mother's milk, the colostrum. And then there's artificial passive immunity where you actually get an injection of an antibody into the bloodstream, and it lasts only as long as that antibody dose is circulating. And so you might get, say, 2 or 3 weeks of resistance against a particular disease. Okay, this protection ends when the antibodies naturally break down. And so here you can see sort of a flow chart showing you these different types of immunity. This is a flow chart dealing with humoral immunity. Humoral immunity, remember, is antibody-mediated immunity. In active immunity, what happens is that the body actually produces the antibodies against the antigen, and this is something that lasts your entire life. If it's naturally acquired, it comes as a result of activities of daily living. If it's artificially acquired, you get it through a vaccine. In passive immunity, what happens is that you basically rent antibodies out from another source for a short block of time. Naturally acquired passive immunity we find in mother's milk to the infant, and also as a result of placenta delivering mother's antibodies to the fetal circulation, whereas in artificially acquired passive immunity, you get an injection of antibody into the bloodstream, and that's only going to last until those antibodies are turned over, they're attenuated. Okay, that brings us to the end of this block, and I'll see everybody in class. Okay, we want to close out Chapter 21 now and talk about innate and adaptive body defenses. In particular, we're going to start out talking about antibodies. Now, what is an antibody? An antibody, for lack of a better term, is what I would call a magic bullet. Essentially what they are protein chains, too heavy and too light, making up the basic antibody structure, that feature two antigen binding sites per antibody, and these are encoded by the DNA within the lymphocyte, and they're going to be specific for one particular antigen. These are generally found in the blood plasma. They're secreted by plasma cells, which are derivatives of B cells, and they're capable of binding specifically with that particular antigen. So again, one antibody, one particular antigen. And they're grouped into one of 5 Ig classes based on the composition of what we call the heavy chain. Okay, so here you can see the basic structure of an antibody. The light chains shown here, the heavy chains shown here, and essentially the composition of the constant regions, as indicated by here the light blue and the light purple, don't vary within an antibody class from antibody to antibody. They remain constant. However, the antigen binding sites, indicated here by the dark purple and dark blue at the ends of this Y-shaped molecule, are encoded by any of a combination of different gene segments that ends up producing now a gene product that's capable of encoding an entire polypeptide chain. So the business end of the molecule is here. The division into different classes depends on the composition of the constant chain. These are Y-shaped antibody monomers. They have 4 looping polypeptide chains. These yellow regions here are disulfide bonds. They have 2 identical heavy chains with a hinge region and 2 identical light chains. So here again are the light chains, here again are the heavy chains. The variable region at one end of each arm combine to form 2 identical antigen binding sites. The constant regions of the stem determine the antibody class, and these classes are IgM, IgA, IgD, IgG, or IgE. And they serve common functions in all antibodies by dictating the cells and chemicals that the antibody can recognize. And it also dictates how the antibody functions to eliminate antigens. Basically, the different class determines where you will find each of these different antibodies. And so here you can see an example of each of these classes. IgMs are pentamers. They tend to be larger than other antibodies, and they are the first antibody that's released upon exposure to an antigen. They're very potent at agglutination. Agglutination is essentially another term for immunoprecipitation. What happens is that it binds to the antigen, and as it continues to bind antigen, all these binding sites become occupied, and then more pentamer moves in to bind other antigens via their binding sites, and eventually what happens is that we generate now a large molecular weight complex that falls out of solution and becomes an easy target for a wandering macrophage. IgA can be a monomer or a dimer, and it's found in mucus and in other secretions, and it helps prevent the entry of pathogens. And so here you can see that the fact that this is a pentamer and that this is a dimer is determined in large part by the composition of the constant region. The antigen binding region always serves the same purpose. It binds the antigen on the invading pathogen. IgDs are monomers that are attached to the surface of B cells, and they function as B cell receptors, while IgGs are monomers. They make up about 85% of the antibodies in plasma, and they come from secondary and late primary responses. These antibodies are small enough to cross the placental barrier, and there are some very important considerations regarding that phenomenon, the fact that IgG can cross. IgE is a monomer that's active in some allergies and parasitic infections, and it causes mast cells and basophils to release histamine. B cells can switch antibody class but retain their antigen specificity. Initially, they generate IgMs, which are capable of binding the antigen, but they're less effective at knocking the pathogen out, and then they switch to IgG later. Almost all secondary responses are IgG. Antibodies inactivate and tag antigens, but don't destroy them per se. They form antigen-antibody complexes. Defensive mechanisms used by antibodies include neutralization, where we essentially cover up the toxic component of the antigen, and agglutination, where essentially what happens is that we cause the production of a large molecular weight complex that immobilizes the pathogen. And this can result in precipitation. And another important aspect to this defense mechanism is the fixation of complement. Remember that complement is a series of proteins that are part of the nonspecific branch of immunity, which aid in the processes that we associate with inflammation. They help with phagocytosis, they help with the inflammatory response, they attract macrophages, and they also form membrane attack complexes that cause the cytoplasm of the targeted pathogen to leak out and the pathogen to die. Neutralization is the simplest defense. The antibodies block sites on viruses or bacterial exotoxins and prevent them from binding to receptors on tissue cells and causing the illness. As a result, the antigen-antibody complexes can be phagocytized and removed from the body. Agglutination is different. Here, antibodies bind the same determinant on more than one cell-bound antigen, and this cross-linked anti-antibody complex agglutinates, and this results in the clumping of the particular pathogen. Eventually what happens is that this entire complex is phagocytized and destroyed. Precipitation is where soluble molecules, as opposed to living cells, are cross-linked. The complexes precipitate and are subject to phagocytosis, just as in agglutination. Complement fixation is a very important auxiliary arm of nonspecific immunity that relies on specific immunity in order to function. It's the main antibody defense against cellular antigens or invading pathogens, such as bacteria, viruses, and so forth. Several antigens can be bound by antibodies and close together, forming a complement binding site on the stem region. So essentially what happens here is that the antigen binding site will interact with the antigen. The constant region will be exposed, and it can now bind to complement. What happens as a result of complement fixation is that the cell is given a biochemical signal to destroy itself, so the cell will lyse. Activated complement functions in amplifying the inflammatory response, in promoting phagocytosis via opsonization, and it also accelerates a positive feedback cycle that causes more and more defensive elements to come essentially to the location of the pathogen and destroy it. Another mechanism whereby complement destroys cells, while we're on the topic of cell lysis, again, is the formation of the membrane attack complex. Remember that the other complement proteins will form around C3b, generating a pore in the cell membrane of the invading pathogen or infected cell and cause the cytoplasm to leak out. Okay, so two methods here whereby we can trigger cell destruction. And here's just a flowchart showing you how these antigen-antibody complexes interact to eventually result in the destruction of the pathogen. Okay, so here we've got our invading antigen. Here's our antibody, and it can be inactivated by neutralization, agglutination, or precipitation, all of which are illustrated here. Remember that neutralization and agglutination involve primarily some sort of living tissue, okay, whether it be a bacterial cell, a foreign cell, such as a protozoan, or in this case a mismatched blood transfusion, for instance, or a virus particle, which we could consider in some ways to be a viable living pathogen. Well, precipitation primarily is aimed at antigens that are on the presence are present on the surface of toxins, basically compounds that cause disease, as opposed to living entities. Complement, of course, is triggered again by the binding of C3b and the formation of the membrane attack complex as a result of antibodies binding to antigen on the surface of the cell to be targeted, and this results in cell lysis. It also enhances the inflammatory process, and it also draws macrophages to the area, stimulating phagocytosis. So all of these work hand in hand to help us eliminate the pathogen. Okay, scientists have used monoclonal antibodies as clinical research tools. Monoclonal antibody is essentially an antibody that recognizes only one particular antigen, so it can be used as a molecular tag to investigate where specific molecules are produced in living systems and what role they might have. This is an antibody that's commercially prepared for a single antigenic determinant, and it's generated by a cell type called a hybridoma, which is essentially a fusion of a tumor cell with a B cell. And what happens as a result is you make an immortal B cell that's capable of generating antibody as long as that cell culture is provided oxygen and nutrients. And so the antibody can be harvested, purified, and then be used in combination with, say, fluorescent dyes that can be attached to the constant region in order to investigate where particular molecules might show up in living tissues or cells, and it could give a clue as to their potential role. These cell lines proliferate indefinitely, they're immortal, and they have the ability to produce a single type of antibody, and again, we use them in clinical research. Antigen-antibody complexes don't destroy the antigens, but they prepare them for destruction by our innate defenses. The antibodies don't invade solid tissue unless a lesion is present. They can act intracellularly if they're attached to virus before it enters the cell, and this can activate mechanisms that destroy the virus. So it can endocytose, hitching a ride on a virus particle, and that can in turn trigger reactions similar to those triggered by interferon. So let's talk about cell-mediated immunity for a bit. T cells provide defenses against intracellular antigens. Some T cells directly kill cells while others release chemicals that regulate the immune response. So T cells come in three flavors. Okay, there's helper T cells, which stimulate the humoral immunity and stimulate cell-mediated immunity. There are cytotoxic T cells, which are stimulated by helper Ts and then actually show up where the diseased tissue is, recognize the foreign antigen display, and then destroy that cell, target it specifically for destruction. And then there's regulatory T cells, which are essentially the off switch for the response that wipes out the pathogen. So all three of these are educated in the thymus. Two populations of T cells are based on which glycoprotein surface receptors they display. CD4 cells usually become helper Ts, which activate B and T cells, as well as macrophages, and direct the adaptive immune response. Some of these become regulatory T cells, which turn the immune response off, and some can also become memory Ts. CD8 populations become cytotoxic Ts. These destroy cells harboring foreign antigens, and they also become memory Ts during the course of the infection. So the next time that same strain of pathogen shows up, there's a vast population of cytotoxic Ts as well as B cells waiting to battle the disease. Helper, cytotoxic, and regulatory Ts are all forms of activated T cells. Naive T cells are simply termed either CD4 or CD8. These are cells that are not immunocompetent and have not experienced antigens, so they haven't differentiated and fought disease. Naive can also refer to CD4 or CD8 cells which are in a state of what's called anergy, where they've been rendered essentially um, living but inert, incapable of mediating an immune response during the process of positive and negative selection that generates all the lymphocytes that make up the immune system. And so this is just a chart showing you the major types of T cells and what they do. Okay, here's the immature lymphocyte in the red marrow that migrates here to the thymus. Eventually the CD4 line is going to be active as a memory cell or an actual helper T or regulatory T. And over here, this other type of cell, CD8, is going to eventually mature and it will become activated upon exposure to antigen, and these will become the cytotoxic Ts that actually show up where the disease tissue is located based on the antigen display and destroy that particular tissue. T cells respond only to processed fragments of antigens that are displayed on the surfaces of cells in combination with a protein called MHC. Antigen presentation is vital for the activation of naive T cells and the normal functioning of effector Ts. Two types of MHC are important to T cell activation. One are class 1 MHC proteins, which are displayed on every single cell in the body except for erythrocytes, and the other are class 2 MHCs, which are displayed by antigen-presenting cells. These include macrophages, dendritic cells, and B cells. And what you have to understand about the MHC is its job is to do what? Its job is to present an antigen on the surface of that cell to alert the immune system to the presence of a foreign antigen, and as a result, the potential exposure to a pathogen, or to identify a cell as belonging to your own body's tissues, and as a result, uh, mark it as off limits for attack by the immune system. So they're both sort of like hors d'oeuvre trays, with the antigen being the hors d'oeuvres. Both types are synthesized by the endoplasmic reticulum, and they bind to peptide fragments. Class 1 MHCs bind with fragments of protein synthesized inside the cell, so these are what are called endogenous antigens, which are self-antigens in a normal cell and a non-self antigen in an infected cell. They're crucial for CD8 activation. They inform cytotoxic Ts of microbes hiding inside the cell, and they act as antigen holders for the self part of the T cell that is recognized as being part of our own tissue. Class 2 MHCs have a similar function, but they're found on different cell types. They bind with fragments of exogenous antigens that have been engulfed and broken down by a phagosome, and they're recognized by helper Ts, and they signal CD4 cells that help is required. So this is the method whereby the helper T is capable of stimulating the cytotoxic Ts and the Bs. CD4 and CD8 cells have different requirements for MHC protein, which presents antigens to them. CD4 cells that become T helpers bind only class 2 MHCs that you find on antigen-presenting cells. Again, the macrophages, the dendritic cells, and the B cells, and the cytotoxic Ts. CD8 cells become cytotoxic Ts and bind only class 1 MHC proteins on antigen-presenting cell surfaces. Once they become activated, cytotoxic Ts seek the same antigen on a class 1 MHC protein that's displayed on an infected cell, which is what you'd expect. CD cells are activated by class 1 MHC proteins. The question is, how do antigen-presenting cells get endogenous antigens from another cell and display them on class 1 MHCs? Well, dendritic cells can engulf dying virus, infected or tumorous cells, or import antigens through temporary gap junctions with infected cells and then display them both on class 1 and class 2 MHCs. And this serves again as the warning that there's a stranger in town. And so this is just a table. This is 21.5 in your book. And what it's showing you are the class 1 and class 2 MHCs, the type of cells that display them, the type of cells that recognize them, the foreign antigens on MHC, where they come from, and the foreign antigens on MHC which signal a particular cell population. So this is a good summary slide here, so I would star this because test material will come from the information contained on this slide. T-cell activation is a two-step process. It involves antigen binding and co-stimulation, and both occur on the surface of the same antigen-presenting cell, and both are required for clonal selection. T-cell antigen receptors bind to antigen MHC complexes on APCs, and these receptors recognize the non-self-self complex linked to multiple intracellular signaling pathways. So what happens then once the recognition occurs is that we turn on a signaling pathway inside the cell that's going to render it now capable of attacking anything that displays that foreign antigen on an MHC. Other T cell surface proteins involved in T cell activation help maintain coupling during antigen recognition. Co-stimulation requires T cell binding to other surface receptors on the antigen presenting cell to generate co-stimulation. Dendritic cells and macrophages produced surface B7 proteins when innate defenses are mobilized, and B7 bind crucial co-stimulatory signals. Cytokines are essentially proteins that signal different cell populations to begin mitosis and become competent. So they trigger proliferation and differentiation of the activated cell. Without co-stimulation, this particular cell population will go into anergy. The T cells will become tolerant to the antigen. They'll be unable to divide, and they'll fail to secrete cytokines. So they'll basically be out of the picture. T cells that are activated enlarge and proliferate in response to the cytokine signal and differentiate and perform functions according to their class. And so here you can see an example of how this operates. What you've got here is a dendritic cell with an antigen display on a class 2 MHC. So what happens now is that the T cell with the appropriate receptor is going to recognize this antigen display. Okay, this is T cell receptor antigen MHC recognition along with co-stimulation as a result, again, of ancillary proteins that are also on the APC surface. And so what happens here is that this particular CD4 cell is going to become activated. Okay, once this occurs, once it's become activated, it's going to generate many copies of itself by going into differentiation and cell division. Some of these cells will become memory Ts, and the rest will, in this case, become helper Ts. And what the helper Ts will do is stimulate now the humoral and cell-mediated arms of immunity via the production of other interleukins that are designed to accelerate now this response. So without the helper T, we lose a critical point in this cascade, this signaling cascade, that energizes the B and the cytotoxic T cells to actually get to work. And this is why diseases that affect T cell count affect immunocompetence. Okay, so let's look at proliferation and differentiation. The primary T response peaks within a week. T cell apoptosis occurs between days 7 and 30. The benefit of this is that activated Ts can be a hazard. They can produce large amounts of inflammatory cytokines which can trigger hyperplasia and cancer. And so we only want them around during the time that they're fighting the infection. Once it's over, we need to turn them off. Effector activity wanes as the amount of antigen declines, and the amount of antigen declines as the attack is successful. The memory Ts will remain behind and mediate secondary responses should that antigen show up again. Cytokines are chemical messengers of the immune system, and they mediate cell development, differentiation, and response in the immune system, and these include interferons, which are defenses against viral infection, and interleukins, which are signals that are sent between white blood cells. This is what interleukin literally means, which stimulate the proliferation and differentiation of B and T cell populations. Interleukin 1 is released by macrophages and co-stimulates bound T cells to release interleukin 2 and to synthesize more IL-2 receptors. IL-2 is a key growth factor, and it acts on cells that release it and on other T cells, and this encourages the activated T cells to divide rapidly and to differentiate. So this is what generates now our army that we're going to use to fight the invading pathogen. Other cytokines amplify and regulate innate and adaptive responses, such as tumor necrosis factor, which is a cell toxin that destroys infected cells, and gamma interferon that enhances the killing power of macrophages so that they can destroy now infected cells or cell debris. The helper T is pivotal in all this. It plays a central role in the adaptive immune response. It activates both the humoral and the cell-mediated immunity, and it does this by the production of these interleukins. Once it's been primed by the antigen-presenting cell, it activates the T and B cells and induces their proliferation. Their cytokines also recruit other immune cells with the same specificity so that we can effectively fight the disease. Without the T helper cells, the immune response is very weak. These helper Ts interact directly with B cells displaying antigen fragments that are bound to MHC2s, and they stimulate them to divide more rapidly and begin antibody formation. The B cells may be activated without T helpers by binding to T cell independent antigens, but this response is relatively weak. Most antigens require T helper co-stimulation to activate B cells, and these are what are called T cell dependent antigens. And so you can see here how the T cell does its job, right? First, we have the T helper cell binding with the self-non-self complex that's displayed by the B cell. And again, one receptor recognizes one particular antigen display. So the T helper that's going to be energized and the B cell that's going to be selected are going to be the ones with this particular specificity. And so what happens is that it complexes with the B cell, and as a result of this interaction, the helper can release interleukins that stimulate the B cell to complete its activation and to begin to divide. Okay, so here you can see all the different components. This is the helper T, this is the B cell. Okay, and what you're seeing here is the T cell receptor. This is the antigen that's being displayed by this particular B cell, okay, this little red guy here. And this is the MHC2 protein upon which the antigen sits. So the T cell receptor recognizes this display. It's this recognition that's going to trigger now the release of interleukins that's going to stimulate this B cell to differentiate, divide and differentiate. There's also CD4 protein that comes into play here, okay, that helps with the recognition. And here you can see the interleukin that's being produced, in this case IL-4, okay, and the B cell goes off and does its job. CD8 cells require T helper activation to become destructive cytotoxic Ts, and they cause dendritic cells to express co-stimulatory molecules that are required for CD8 activation. And so what you're looking at here is essentially the same thing, okay, except that we use the APC as an intermediary. The antigen presenting cell engulfs the infectious agent and displays it on its surface. Okay, what happens as a result is that the T cell can recognize this antigen display, and CD4 plays a part in this, right? This is again a class 2 MHC because this here is a macrophage. Now what happens as a result of this helper T has been activated. He's going to produce interleukins, in this case IL-2. The CD8 cell also recognizes a class 1 MHC antigen display. Okay, the specificity is the same. Okay, the antigen is being picked up as a result of this interaction. Okay, and its proximity to the helper T the CD8 cell is going to become an activated cytotoxic T, and he's going to march off and destroy any tissue that has this same antigen display. Okay, if you're showing this to me, this means that you need to be destroyed. So what happens is this guy will begin to divide and differentiate and go off and fight our battle for us. So what all do helper T cells do? Well, they amplify innate defenses by activating macrophages and causing them to become more potent killers, and they help mobilize lymphocytes and macrophages and attract other types of white cells to activate them so that they can be more effective at battling the infection. There's different subsets of helper Ts. T helper 1 cells mediate most aspects of cellular immunity, while helper 2s defend against parasitic worms, they immobilize eosinophils, and they promote the allergic response. And this is why a differential count in which your eosinophil percent is increased indicates either a parasitic infection or an allergic response. Helper 17 cells link adaptive and innate immunity by releasing interleukin 17, and this plays a role in autoimmune response. Okay, remember that we talked about the fact that occasionally what can happen is the immune system can turn around and attack your own tissue. And so what occasionally happens here is that you can have now a population of B or T cells that suddenly recognize and interact with your body's own antigen display, which shouldn't normally happen. And we think that part of the way that this happens is that we can remove some of the regulation that keeps the cells that would otherwise recognize and destroy your own tissues in a state of basically unconsciousness. And so they can turn around then and attack your own tissue. Also, exposure to cross-reacting antigens is the other prevailing theory regarding how this autoimmunity can come about. Cytotoxic Ts directly attack and kill other cells. The activated cytotoxics circulate in the blood and lymph as well as the organs, looking for body cells that display that particular antigen so they can destroy them. Some of the targets of cytotoxic Ts are virus-infected cells, cells that have bacteria or parasites inside them, cancer cells, and foreign cells that can come as a result of a transplant or a transfusion, just as a couple of examples. It can destroy all infected or abnormal cells. The lethal hit from a cytotoxic T comes by two different methods. The cytotoxic T can release perforins and granzymes by exocytosis. The perforins basically punch holes through the cell membrane of the infected cell, causing the cytoplasm to leak out and the cell to die. While the granzymes are different. The granzymes will trigger that apoptotic switch that I was talking about earlier, which is the suicide switch that tells a cell to kill itself. The cytotoxic T cell binds specific membrane receptors on the target cell, and this can stimulate apoptosis as well as production of these punching holes in the membrane of the infected cell. And here you can see this in action. Okay, so we can see on the left here the series of events that results in the attack and destruction of a target. Here the cytotoxic T IDs the foreign antigen on an MHC protein and binds tightly to the target. The next thing that happens is it releases perforin and granzymes. The perforin punches holes in the cell membrane, causing the cytoplasm to leak out, while the granzymes trigger the apoptotic response. And so eventually what happens is that the targeted cell is going to be destroyed. After it does its job, the cytotoxic T detaches and looks for another target, so it can destroy more than one infected cell in the course of doing its job. That's why it's so effective. Natural killers are a little different. They recognize other signs of abnormality that indicate that a cell is infected. They don't have class 1 MHC. They have antibodies that coat the target cell, and they have a different surface marker that they look for on cells that are displaying signs of infection. But they use the same mechanisms as cytotoxic keys to kill their target. They produce perforins and they produce granzymes that ultimately result in the cell destruction. These guys are responsible for a phenomenon in the immune system called immune surveillance. The natural killers and the cytotoxic Ts basically walk the beat of the entire body looking for any cell that's gone rogue, that's gone cancerous, and they'll destroy it. With a suppressed immune system, these random events that happen 2 or 3 times a day, we get cancer, are not going to be caught and destroyed, and as a result, these abnormal cells can eventually become metastatic and spread throughout the body. And this is why people with suppressed immune systems not only have increased incidence of infection, but also increased incidence of cancer. The T regulatory cells are the off switch for this entire process. They dampen the immune response by direct contact or by inhibitory cytokines such as IL-10 or TGF-beta. TGF stands for tissue growth factor beta. They're important in preventing autoimmune reactions by suppressing self-reactive lymphocytes in the periphery. We've been doing a lot of work on these regulatory cells to use them to induce tolerance for transplanted tissue so that we can broaden the potential universe of donor tissue for people that need life-saving organs in order to continue to live. And this is just a flowchart showing you a summary of the primary immune response. This is the whole ball of wax. So we've kind of already gone through most of this. But again, this is a good summary slide. Okay, I would star this. This talks about complement. Okay, this talks about the role of the B and the T cells and how they do their job. And it talks about how the helper T is a critical link between these two universes, right? Because it produces the cytokines that get the B and the cytotoxic Ts into gear so that they can fight the invader. And it talks about the production of memory cells on both ends of this response so that the next time that same pathogen shows up, we can effectively destroy it without getting sick. Okay, organ transplants come in 4 varieties: autografts, isografts, allografts, and xenografts. An autograft is tissue that's transplanted from one body site to another in the same person. An isograft can only happen between identical twins, and an allograft are between individuals who are not identical twins. A xenograft is tissue that comes from another animal species. Now you might think, my God, how would allografts and xenografts ever potentially work? And the success of all of these depends on the similarity between the two tissue types. Autografts and isografts are generally used for donor tissue. They're almost always successful if there's a good blood supply and no infection, because there's no difference in the antigen display, right? If you graft your own tissue onto your own injury, it's going to take, right? Your immune system is not going to recognize that as something alien and destroy it. An isograft is the same thing, right? Because of an identical twin, the MHC loci are going to be the same because they're genetically identical. As a result, the graft of tissue from one identical twin to another should not result in rejection either. So if you're an identical twin, you're lucky because you were born with an organ bank that will always be a match. Xenografts are something that's kind of new. What we're trying to do is express human antigens on genetically engineered animals so that we can have donor tissue that comes from something other than human that can manage to take. We've already done this by generating transgenic pigs in an effort to use skin grafts or even heart valve transplants, for instance, as a way of attempting to fix these kind of defects in humans. And we've had some success. Most common, however, is the allograft. And what you have to do there is find somebody who's a tissue match. And generally, that comes from a close relative. If not, you have to get lucky in the sense that you have to have somebody accidentally die who happens to be a tissue match for you. ABO and other blood antigens, as well as MHC antigens, need to be matched as closely as possible in every case except for those tissues that have immune privilege. Okay? Now remember, those are the liver, the corneas of your eyes, the tissue of your central nervous system, and the gametes that are present inside the male and female duct system. After surgery, the patient is usually treated with anti-inflammatories and immunosuppressants, and this is an effort to try and get the graft to take. Many of these will have severe side effects, unfortunately. The patient's immune system is suppressed. As a result, they're more prone to infections, and this can lead to increased mortality. Most balanced drugs against graft survival with minimum toxicity, and we try and use antibiotics to control bacterial infection. Best circumstance is rejection after 10 years in 50% of patients. So the flip side of this is that organ donors are really doing a marvelous service to mankind, because in the event of your accidental death or your natural death, the likelihood that your organs will save somebody who is on the transplant list is very high, whereas the reverse is a much more difficult proposition. The idea that you need an organ and somebody out there who is a tissue match is going to accidentally die within the window that you need the organ can be very remote. So we've done research to try and induce tolerance. We can try and suppress the recipient's bone marrow or douse the recipient's marrow with bone marrow from the donor of the new organ, and this combined immune system may treat the transplanted organ as belonging to yourself, but that's not always a guarantee. And we hope to harness regulatory T cells in order to suppress immune reactions. This is all, again, cutting-edge research. Immunodeficiencies are essentially malfunctions in the immune system that can either be inherited or acquired. They can impair the function or production of immune cells or molecules such as complement, or the production of antibodies, or production of T cell receptors. Any of these are possible. SCIDS is an example of this. This is an inherited disorder that affects the lymphocytes. We have a deficit in B and T cells because we have a defective ADA gene. This produces metabolites that are lethal to T cells and causes them to die. It's fatal if it's untreated. We generally try and treat it with bone marrow transplants, but this is the classic example of the boy in the plastic bubble. These individuals have to live in a sterile environment until and or if they're capable of getting a transplant that can save their lives. Other than that, any infection can lead to death because they have no method, no active method of defending themselves against disease. Hodgkin's is an acquired immunodeficiency. It's basically a cancer of the B cells, and it leads to immunodeficiency by depressing lymph node cells and flooding the system with lymphocytes that are incapable of fighting disease. And so what happens is that even if you have immunoconfident lymphocytes, their ability to do their job is impaired by all of these non-specific white cells that are basically clogging up the system. AIDS is another acquired immunodeficiency. It cripples the immune system by interfering with the activity of helper T's. It's characterized by weight loss, night sweats, and swollen lymph nodes, and an increase in opportunistic infection and opportunistic cancer. And the reason for this, again, is that not only have we suppressed now the ability to fight pathogens because the T cells aren't doing their job, they're essentially decreasing in number, and in addition to that, the T cells are being transformed into virus factories, which not only fail to help the immune system, but they also display antigens on their surface that are suicide pills for other lymphocytes that they interact with. And so it's a double whammy here. But we suppress also the immune function, the immune surveillance function of the immune system. So we're not as good at fighting incoming pathogens. We also don't check the body adequately for the everyday mistakes that result in cancerous tissue appearing spontaneously in tissues that are mitotically active. And so this is why you see an increase in opportunistic cancer and infection in these individuals. It's caused by the HIV virus, which is transmitted by body fluids such as blood, semen, and vaginal secretion. HIV enters the body through blood transfusions, blood-contaminated needles, or sexual intercourse, oral sex. It can also be transmitted from mother to fetus. And what it does is it destroys the helper T population and suppresses cell-mediated and humoral immunity. Um, Generally, HIV multiplies in lymph nodes during its asymptomatic period. This can go on for a decade if it's not treated. The symptoms occur when the immune system finally collapses. The virus can invade the brain, leading to dementia, and HIV-coated glycoprotein complexes can attach to the CD4 receptor. This is how the virus gains entry into the helper T and uses reverse transcriptase to generate its genetic material. As a result, it's what's called a retrovirus. The DNA copy directs the host cell to make viral RNA and protein and enables the virus to reproduce. So we transform the helper T from something that mediates our humoral and cell-mediated immunity into something that is a virus factory. So these extra particles go out and infect more helper Ts. In addition to that, the infected helper Ts kill the lymphocytes that they come into contact with. And so your white cell count starts to drop precipitously. Reverse transcriptase is an error-prone enzyme that is unique to the retrovirus family. As a result, it presents a drug target. So we can treat with antiviral drugs. AZT was one of the earliest drugs used to try and inhibit the ability of reverse transcriptase to do its job. There are other drugs that we can use in the form of a cocktail in order to basically turn this once lethal condition into a chronic disease. As an example, we have fusion inhibitors that can block HIV's entry into the cell, integrase inhibitors that block viral RNA integration into the host DNA, and reverse transcriptase and protease inhibitors that inhibit viral replication enzymes. In addition to that, I guess I would put AZT in this third category here. AZT is essentially a toxic building block that reverse transcriptase doesn't recognize as being an abnormal nucleotide, and as it incorporates it into the DNA, essentially what happens is that the retroviral genome can be completed, and this results eventually in the production of virus particles that have no genetic material, or genetic material with so many mistakes that it's not viable. Antiretroviral vaginal gel can reduce the risk of contracting AIDS after intercourse with an infected partner by up to 50%. With these, now, before we move on to autoimmunity, a note here, okay? We talked about the modes of transmission, and it's through body fluids, intravenous drug use, and transfusions that are contaminated, although that's very rare anymore in the United States because now we routinely test donor blood for the presence of HIV, and obviously if you have HIV you cannot be a blood donor, so the incidence of that has gone way down. There's transmission via sexual intercourse, whether it be same-sex sexual intercourse or heterosexual intercourse, because of the potential for infected secretions to mix with the blood. Okay, this is much more frequent in same-sex sexual intercourse than it is in heterosexual intercourse, hence the difference in the rate of transmission. But we should note something here, which is that viruses, as bacteria, as protozoans, as any pathogen can do, can mutate. All right, so when we say that it can only be transmitted by these methods, we should always put the qualifier in at this time, okay? Because it's possible that the virus could acquire a method of transmission that wasn't previously anticipated as a result of acquiring or changing its genetic material and essentially providing now a new pathway to infect previously individuals who are not at risk. Okay. Okay, autoimmune disorders. This is where the immune system loses its ability to distinguish self from foreign antigens. We generate autoantibodies and sensitize cytotoxic Ts that destroy body tissues. Examples of these conditions include myasthenia gravis, Graves' disease, type 1 diabetes, systemic lupus, glomerulonephritis, and rheumatoid arthritis. Essentially, there are different proposed mechanisms that can lead to autoimmunity. Okay, we can suppress the entire immune system. We can have anti-inflammatory drugs such as corticosteroids that block cytokine action and block co-stimulatory molecules that can lead to autoimmunity. We've done a lot of research into the activating of regulatory T cells, including self-tolerance, using vaccines as a treatment for these conditions, as well as directing antibodies against the self-reactive immune cells. Mechanisms for autoimmunity. Weakly self-reactive lymphocytes can be activated by foreign antigens that resemble the self-antigens. This is, again, the cross-reacting antigen theory. These antibodies against foreign antigens can cross-react with your own tissue. New self-antigens can also appear. They can be generated by gene mutations or changes in the self-antigens by hapten attachment or infectious damage, or the release of novel self-antigens by trauma to a barrier, such as the blood-brain barrier, the blood-testis barrier. Okay? And also, we should note, by genetic mutation in the lymphatic system itself, okay, where it causes you to generate an abnormal antibody or T cell receptor. Okay, all of these are possibilities. Hypersensitivity is basically where the immune system overreacts to exposure to an antigen. The immune responses to the perceived threat cause tissue damage. Different types can be distinguished by the time course and whether antibodies or T cells are involved. Antibodies can cause immediate and subacute hypersensitivities, and T cells can cause delayed hypersensitivity. Immediate hypersensitivity can be classified as type 1, which begins seconds after contact with the antigen. The initial contact is asymptomatic, but sensitizes the person. The reaction can be local or systemic. The mechanism involves IL-4 secreted by helper T2 cells. The IL-4 stimulates the B cells to generate IgE, which binds to mast cells and causes basophils to flood the body with histamine, and this initiates the inflammatory response. A later encounter with the same antigen will generate the allergic reaction. Allergic reactions can be local or systemic. If they're local, the mast cells of the skin and the respiratory tract and the gastrointestinal mucosa are involved. The release of these histamines can cause dilation of blood vessels, leading to runny nose, hives, watery eyes, heat, pain, itching. This can result in asthma if the allergen is inhaled. We can use antihistamines to control this, or even corticosteroids. A systemic response that's severe enough is classified as anaphylactic shock and can result in death. This is a response to the allergen that directly enters the blood and circulates rapidly. The basophils and the mast cells are enlisted throughout the body. Systemic histamine release can cause constriction of the bronchioles, swelling of the tongue. Sudden vasodilation and fluid loss from the blood can cause circulatory collapse and even death. The treatment is epinephrine because epinephrine is an anti-inflammatory. And so what you're looking at here is just the mechanism of an acute allergic reaction and the response. Subacute hypersensitivities are caused by IgM or IgG transferred by plasma or serum. They have slow onset and long duration. Cytotoxic reaction, where the antibodies bind to antigens on a specific body cell and stimulates phagocytosis and complement-mediated lysis of cellular antigens. Examples of this include mismatched blood transfusion reactions. Type 3 hypersensitivity is different. These antigens are widely distributed in the body or the blood, and insoluble antigen-antibody complexes form that can't be cleared from an area of the body, resulting in inflammation, cell lysis, and cell killing by neutrophils. An example of this is a condition known as systemic lupus erythematosus. Delayed hypersensitivity, classified as type 4, slow onset. The mechanism depends on the helper T cells involved, and cytokine-activated macrophages and cytotoxic Ts are the ones that cause the damage. An example is allergic contact dermatitis, such as exposure to poison ivy or poison oak. The antigens act as haptens, and what happens is that once they associate now with your body's own tissues, the hapten can be recognized as an antigen, and as a result, we can generate now the traditional T and B cell response. TB skin test is one of the ways that we check for delayed hypersensitivity to the tuberculin antigen. Okay, what follows here are some videos that sort of go into a little more detail regarding these different types of hypersensitivities.
Some people develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented to T helper cells. Tissues under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines, which stimulate B cells in the area to proliferate and differentiate into IgE-producing plasma cells. As IgE is produced in specific areas of the body, the IgE molecules attach to nearby mast cells. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen immediately binds to the IgE antibodies, which are already attached to the mast cell. Within seconds of this binding, the mast cell releases histamine and other chemicals that cause an inflammatory response. These chemicals trigger a variety of symptoms such as capillary dilation, airway constriction, mucus secretion, pain, and itching. Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups, based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients. Immune complexes consist of antigens and antibodies bound together. Although large immune complexes are engulfed by phagocytes and removed, intermediate-sized immune complexes are not readily removed. These intermediate complexes react with and activate complement. For example, complement activation in blood vessels causes basophils to degranulate, which causes vasodilation. Immune complexes entrapped in blood vessels or other tissues, such as kidney glomeruli, activate complement, which attracts neutrophils and causes them to degranulate. Neutrophils release enzymes responsible for tissue damage. Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TD cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T-cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
Okay. How is the immune system formed? Where does it come from? The immune system cells develop in the liver and the spleen between weeks 1 to 9. The bone marrow becomes a primary source of stem cells later and throughout adult life. Lymphocyte development continues in the bone marrow and in the thymus. T helper 2 lymphocytes predominate in the newborn, while T helper 1s are basically trained in the thymus as the person encounters antigens. Influences on immune system function. Nervous system malfunctions related to the immune system can lead to depression, emotional stress and grief, which can impair the immune response. Okay, this is how these systems can interact. Your diet can influence the immune response. Vitamin D, for instance, is required for activation of CD8 cells into cytotoxic Ts. As a result, vitamin D supplements result in reduced incidence of infectious disease, and deficiencies are linked to autoimmune disorders such as multiple sclerosis. With age, the immune system function begins to decline. We see greater susceptibility to immunodeficiency and autoimmune disorders, as well as greater incidence of cancer. We don't know why the immune system fails, but it may be due to the atrophy of the thymus and decreased production of naive T and B cells as we age. Okay, that brings us to the end of this chapter. Make sure and review this material, as it will be covered on the final regular exam as well as the comprehensive final. And I will see everybody in class.